name is Faye Orcales. I'm a first-year PhD student in the Biological and Medical Informatics program at UCSF. I'm very interested in the genetics of complex diseases, and so I'm excited to share with you some of the work I did in the Liao lab at UCSF before starting graduate school. In this project, I looked at polygenic risk score associations with psoriasis clinical phenotypes across a multi-ethnic cohort. For those who aren't familiar, psoriasis is an inflammatory skin disease with a polygenic mode of inheritance. This means that there are many genetic variants that cause the disease, and it's hypothesized that if an individual has enough of these associated variants, the scale will tip and they will likely develop psoriasis. But it's difficult to look at many genetic variants individually, so a polygenic risk score is a way to summarize the effect of many variants. Essentially, a higher score means you have a higher genetic predisposition. To calculate these scores, we identified 88 disease-associated SNPs from the largest published GWAS study on psoriasis. We further subdivided the score into an HLA and non-HLA score because of their relevant genetic mechanisms in psoriasis. HLA loci are known to affect antigen presentation, and non-HLA loci generally affect innate and adaptive immune activation. Furthermore, we wanted to explore non-HLA associations as it isn't as well studied in literature as the HLA region. So for a cohort of about 600 patients, we calculated 3 polygenic risk scores, PRS all, no HLA, and HLA for every individual, and ran some tests to see which were correlated with specific clinical phenotypes. As a first test, we saw that increasing PRS all was associated with earlier ages of onset. This means that people with higher scores, they have more disease-associated SNPs, and they're more likely to develop psoriasis earlier than those with lower scores. Then we looked at associations with psoriatic arthritis and saw that patients with confirmed psoriatic arthritis had significantly higher non-HLA scores than patients that did not have it. So it's possible that non-HLA SNPs are driving psoriatic arthritis, particularly in those with European or Asian ancestry. This was an exciting finding as psoriatic arthritis is relatively not well understood. We also looked at associations to body location. One interesting thing we found was that non-HLA SNPs seem to be driving inverse in nail psoriasis. There are also different types of psoriasis that can manifest. For plaque psoriasis, we saw that non-HLA SNPs seem to drive its formation, particularly for those with Asian ethnicity. And interestingly, we saw that HLA SNPs seem to be protective against pustular psoriasis in the combined cohort and protective against erythrodermic psoriasis primarily in the Asian cohort. We also looked at associations to different environmental triggers, comorbidities, and treatment responses. Across the combined cohort, we saw that non-HLA SNPs may be driving skin injury as a psoriasis trigger and metabolic syndrome as a comorbidity. Also, HLA SNPs may be driving a positive response to UVB treatment. And I know that was a really quick run-through of our findings. So to summarize, we saw that polygenic risk scores have significant associations with various psoriasis clinical phenotypes. We also saw that there were instances where different ethnicities had different results. Some associations only appeared for certain ethnic groups. I would like to thank the Liao Lab for their support and mentorship on this project, and thank you to the ARCS Foundation for giving me the opportunity to share this research and for supporting me as a first-year graduate student. And if you are interested in chatting or learning more, please feel free to visit me at poster 131, or you can scan the QR code here. Thank you so much for listening.